First of all, thank you for the opportunity to present today, panel. At Precise, we developed a platform, a 3D biofabrication platform. Maybe more precise, we have a 4D platform because it's not just a 3D printer, but it's a process, a full process. And we're utilizing this platform for ophthalmic tissues at this stage. Just in a glance, we developed the platform where our mindset here is really to have a scalable and repeatable method, supply on demand of a fabrication of human cells, human tissues. Currently, we completed our GLP study with our cornea EK, and we have 2 additional proof of concepts and preclinical studies ongoing. Our GMP facility is up and running. We have a world-leading scientific team with strategic investors. And we have our 4D multimodality, GMP-ready, compliant robotic bioprinter. The way we do it, and this is a process, it's a multidisciplinary approach where this is really what is required to, and then at the end of the day, to bring the tissue to the clinician's hands, where the full approach really looks at the clinical relevance, it looks at the supply and demand, of course, the financial aspects. And here, you can see here, and you'll be able to see the printer here running as I'm speaking. We start any tissue from the biomaterials and from the cells. For example, for the EK, we have the basement membrane made of collagen, where we fabricate it, we give it the mechanical properties, the optical properties, permeability, anything that is required for the specific tissue. And together with the cells, where here we source the cells from donor, and we develop the process of expansion and cryopreservation of the cells. So from one primary cornea, we're able to fabricate today between 400 and 500 new corneas. The bioengineering is essential here because we take the cells and we take the materials, but we have to bring them together to be a functional tissue. We're able here to achieve high cell density, over 3,500 cells per square millimeter, a very thin tissue, 12 micron thick, and ease of use, which I'll present in the next slide. Our GLP study was outstanding. The results you can see here. I'll just focus on 2 points. One of them, the corneal thickness, that we were able to bring it back to the natural thickness from one perspective. And on the right bottom, you can see the endothelial cells. We're sure that these are the human endothelial cells and not the animal cells that essentially are doing the functionality that we required. And you can see here where we have a very nice cell density, 2,500 per square millimeter, and you can see the identity and the potency of the cells in the rabbit. Last but not least is really the ease of use, and you can see here using a standard goiter tube. And this is what I mentioned, the mechanical properties that we give the tissue. It's made of collagen and the endothelial cells. You see it just opens up, and you can see on the right a clear and transparent cornea after 3 weeks. And it's really much easier than a DMEC, where you can see now it just opens up and it's in place. We are at a transition phase now, moving into the clinical study this month, where the primary objective here is to assess the safety and the tolerability. Of course, we're checking also the efficacy. We're doing between 10 and 15 patients in the Rambam Medical Center in Haifa. This is exactly where we want to be at this stage, moving forward toward the clinic. What's next? We have 2 additional indications in the ophthalmology field. We have done also a development in other fields, in cardiology and orthopedics, but we're fully focused now in ophthalmology. And you can see here subretina transplant and an intrastromal lenticule that as I mentioned, we're still at the stage of a proof of concept. We have an amazing team. Together with me, founded the company, Dr. Anthony Atala and Professor Shai Soker, both from the Wake Forest Institute of Regenerative Medicine, one of the leading institutes in the world in the field of tissue engineering. I don't think that I have to introduce the medical advisory board. Everybody here in the room knows them, and our chairman comes from the robotics area, from currently at the Medtronic, before that Mazor Robotics. Summarize, we have a very unique platform, 3D printing, or what we call a 4D process. Our leading program, the EK, which is moving now into the clinic. We have a strong multidisciplinary team from biology, biomaterials, electronics, optics, etc. We have a very strong IP portfolio, over 30 patents, and related to the tissues, related to the manufacturing platform. We have a pipeline of products that definitely we are going to see cell therapies moving into the field of ophthalmology. And post the first in human, we are planning to launch our C round just around the corner. Thank you.
Thanks so much. Okay.